Afternoon and welcome to today's Made by McGill alumni webcast, mRNA vaccines and how they might change the course of medical history. My name is Derek Kassoff, Managing Director of Communications at McGill's Office of University Advancement, and it's Thursday, June 16th, 2022. While all of us have been dealing with COVID-19 and the fallout from the first global pandemic of our lifetimes, we have also been witness to one of the greatest scientific achievements of the last half century, which saw the development of an effective COVID vaccine in under 12 months. And who would have ever guessed that the secret was hiding within our own bodies, a mechanism to program our RNA molecules to protect us from this infection. On today's webcast, we are joined by two McGill graduates who helped usher in this technology through their work at Moderna and Flagship pioneering, as well as a pair of experts from the university who have dedicated their careers to perfecting the science and understanding its applications, not only to COVID and other like-minded viruses, but perhaps to HIV, cancer, and global health more broadly. So without further ado, it is my great pleasure to introduce the members of this star-studded panel. We have with us Noubar Afeyan, who's the founder and CEO of Flagship Pioneering, as well as co-founder and chairman of Moderna. He has a Bachelor of Engineering degree from McGill, and just last month was awarded a second McGill degree, an honorary doctorate of science, in recognition of the significant impact he has made on global health. Welcome. Avak Kahvejian is a general partner at Flagship Pioneering, where he leads a team that invents and launches new therapeutic platforms. He has two biochemistry degrees from McGill, a Bachelor of Science and a PhD, and a long track record of developing and running biotech companies that have developed treatments for cancer, autoimmune disorders, and rare diseases. Welcome, Dr. Kahvejian. Jerry Pelletier is a professor here at McGill in the departments of biochemistry and the Gerald Bronfman Department of Oncology, and is a member of the research team at the Rosalind and Morris Goodman Cancer Institute. His research efforts have long focused on RNA biology, including developing an understanding of what regulates this molecule and how it might be used as a focus of new drug discovery. And finally, Amélie Canel-Vallée is a professor in the Department of Sociology, as well as Epidemiology, Biostatistics, and Occupational Health, and the Canada Research Chair in Policies and Health Inequalities. So welcome to all four of you, and thank you for taking the time to join us today as we try to understand the true significance of mRNA technologies and their implications for global health and their individual well-being. A couple of reminders: if you are watching live today and have questions for any of our panelists, you can send them to us by email at aoc@mcgill.ca, and we'll do our best to address them to our guests. And I should warn our viewers, I know there's a lot out there live today. Many of us are here in Montreal, where we're dealing with a severe thunderstorm warning. Clouds are rolling in, so fingers crossed that the power stays on and the Wi-Fi stays strong, and let's do this. So Dr. Afeyan, perhaps I'll begin with you. So first of all, congratulations once again on that well-deserved honorary degree from your alma mater. But before we get into the story behind Moderna and COVID vaccines, I was wondering if you'd be willing to share a little bit of your personal story with us and tell us how a kid who arrived in Canada as a Civil War refugee ended up at McGill and then on the fast track to becoming one of the most successful life science entrepreneurs of our generation.
Well, first of all, thanks, Derek, for having me. And it's a great pleasure to be talking to former colleagues from McGill. Well, as you said, it's hard for me to define how it is that it happened because there was no grand design, and I could say hard work, but I'd also then have to say a lot of luck along the way as well, as is usually the case. It is the case that I moved to Montreal, and when I was 13 years old, I was in awe of this big metropolitan city, having grown up in Beirut. It was right around the time the Olympics were about to happen, so you could imagine as a teenager kind of being thrust in the middle of this city, going through a massive transformation. I hadn't seen snow before, and so hadn't ridden buses before, so all of these things were quite new. I was quite fortunate, although I should say, vis-a-vis McGill, in that we ended up living at the corner of Peel and Sherbrooke, which was closer to where I ended up going to university than the dorms were. And so I grew up there for many years before actually kind of rolling out of bed and going to class at McGill. So that was quite a strong memory. I grew up essentially in Montreal under the shadows of McGill, and ended up getting drawn into science and then into engineering. And so I did an undergraduate in chemical engineering, and then ended up coming down to Boston to do a PhD, and have led a career of entrepreneurship and innovation since then. And one of the things that we founded and we created ended up having some impact. But at the end of the day, it's the story of lots of different people who've kind of found a way to combine their education and their own curiosity to create new products, new companies.
Wow, that sounds wonderful. And I guess you talk about a combination of lots of people. I guess one of those people is one of our other panelists, Dr. Kahvejian. It sounds like you've got science and engineering in your bloodstream as well. And so how did you parlay your McGill education into a really successful career in the biotech field?
Well, as you know, I studied with Nahum Sonnenberg, who was at the forefront of understanding mRNA sciences and what helps mRNA get translated into protein. Made a number of seminal discoveries in the field, and with Jerry Peltier here, who's on the panel as well. And after studying there, I really wanted to do something in the applied world, taking what I learned from the basic research world, and wanted to enter the world of entrepreneurship. That's when I essentially moved down to Boston and joined a startup that was a flagship startup by the name of Helicos Biosciences, which was really, again, at the forefront of technology in the DNA space, doing single molecule DNA sequencing for the first time ever. And in a short amount of time, in a span of 3 years, the company developed the world's first single molecule DNA sequencer. Through that, I ended up getting more and more involved with Flagship pioneering, which was the founder of Helicos. And through my mRNA background, got roped in by Noubar and Stéphane into the early days of Moderna, brainstorming what we could do with the technology, how we could design the technology. And the rest is history. I've been at Flagship ever since, leading a team to ideate and invent new bioplatform companies and launching those companies, always with an eye towards doing broad range things across many, many therapeutic areas, and thinking about bringing new modalities like mRNA and other RNA technologies and cell therapies and beyond to humankind as a new tool in our armamentarium in medicine.
Wow. So we're going to hear all about that, I think, over the next hour, about how these new technologies are changing how we think about medicine and drug discovery. Before we bring our two McGill professors in, I just want to go back to you, Dr. Afeyan, just with one more question. If we sort of go back to the beginning of this whole COVID pandemic, I'm curious if you remember the moment when you first heard about COVID and its severity, and being the chairman of Moderna, what was your initial reaction? Were you like, I got to get to the lab?
Mm-hmm. Well, there isn't just a moment, because it didn't show up as a pandemic in the first instance. In late 2019, early 2020, there was a couple of reports showing up about a pneumonia-like condition in China. And that's not very unusual, because we get these reports quite frequently, and they'll make it to the press and then they'll disappear, a subset of which end up getting more serious. And in this case, we could hear that it was not associated with corona in the beginning. And I remember quite distinctly, because it happens to coincide with the birthday of my second oldest daughter. I have 4 children, and I happened to be quite unusually having dinner on her birthday. I'm usually never in town. But this one time in 2020, on January 21st, I happened to be at dinner with her, Talene, and I got an urgent text from Stéphane Bancel, who's the CEO of Moderna, who was reaching out from Davos, and he wanted to talk. And so, it was fairly cold. I also remember that, because I kind of walked out of the restaurant with a shirt on. It was probably well below zero here. And we had a discussion, and he basically said, look, the discussions here are intensifying around what this could be. It's not clear. There hadn't been any deaths yet, certainly not outside of China. And even then, I think it was a couple. And but he said, look, people are asking us to add this to our programs. What was interesting was, people around the world didn't know Moderna existed, but Moderna at the time was 700 people. We had 20 drugs and vaccines in human clinical trials, 10 in human trials in vaccines, 10 in drugs. And it was quite a substantial entity. Of course, it was not a big pharmaceutical company, and it had no commercial products, but it had a pretty significant effort. So adding yet another program just like that overnight was not something we were in any position to do. So we had to talk about it as partners. And what we discussed was that even though this was not known to be as serious as it turned out to be, it was this unique moment where we could apply our technology in the one way in which it's incredibly unique, and that is how quickly you can code a new sequence and make a whole new vaccine. And of course, in the regular world, as we've learned since, everybody expects vaccines take 3 to 5 years at a minimum to develop. So the notion that you could do it in 2 days is not a big feature. And so there it stood, right? I mean, it's it was an unused feature of our platform. And we thought, wait a minute, this is a moment, a unique moment where we could take advantage of that capability and respond very quickly to a public health concern and just make the first batches of these things in case it would be needed. And that's how we started. Our very first announcement publicly, I think it was January 22nd, 23rd, was essentially saying we will enter the fray and come up with it only because we want to be safe and not sorry. Well, within weeks, it came to North America. Within a month or a month and a half, it was declared a pandemic. Everything shut down. But by that time, we were weeks from dosing the first patients with our vaccine. It took us 44 days to go from sequence to a dosed human, which a lot of learnings, but could only have been done with mRNA for sure.
Mm-hmm. Right. Oh, that's interesting. What a fascinating story. And this may be a good time to bring in Dr. Pelletier, because obviously it took 44 days, to develop the vaccine. But I mean, this is built on, decades of work around understanding, RNA and mRNA. So Professor Pelletier, maybe I'll bring you in. At the risk of asking you to summarize, 40 years of your laboratory work in like a minute and a half, can you tell us, broadly speaking, what is RNA? What is messenger RNA? And how do these modules function in our bodies? And that create the foundation that allowed companies like Moderna to fast track a vaccine?
Sure. Let me start off by thanking you for the opportunity to be here as well. Let me try to provide some context and give you the Reader's Digest version of 60 years of research that went into this. So mRNA was first discovered in 1961, and I'd say the ensuing 10 to 15 years were spent understanding how the RNA code is deciphered and converted into protein. And these discoveries were so important that a Nobel Prize was actually awarded for them in 1968. And then during the 70s and 80s, what we saw was the elucidation of the rules that dictate how much protein is made from different mRNAs and for how long. And these rules are quite important because they were used later on in the vaccine development to optimize protein output from the mRNA molecules that were used. In the 90s and 2000s, once these rules were elucidated, we then saw research being focused on how do cells manipulate these rules to differentiate, to divide during normal processes, and then how are these rules broken in disease, and can we take advantage of these? So that's the fundamental research. And then in the late 2000s, in 2010, you saw the emergence of Moderna, essentially, who got into the RNA biology space and built a very sophisticated pipeline and amassed enormous expertise around converting that molecule into a new medicine. So that's sort of the Reader's Digest version here. McGill's contribution, I'll step back here a bit, actually starts in the late 60s in the chemistry department, where significant chemical expertise was developed around the building blocks of mRNA. And that expertise remains here at McGill to this date today. In the Department of Biochemistry, we see a second hub of expertise emerging in the late 70s when Dr. Nachum Sonnenberg is hired, and his lab is one of the major labs that contributed to the definition of these rules. I was Nachum's third graduate student. I elucidated some of these in my PhD. And as Avak mentioned, when Avak did his PhD, some of these rules were defined by his PhD studies. Mm-hmm. Why is it such an effective vaccine? I can think of 3 points. One, it's very safe. Unlike vaccines that are delivered by viruses, mRNA cannot damage the genome, and it's not an infectious product. Two, once that mRNA is delivered to the appropriate cells in the body, it produces a lot of protein, so it induces quite a potent immune response. And then lastly, I would say mRNA design is very simple, as you just heard, and it can easily be scaled up. So it's a plug and play technology, right? You get a new sequence within a matter of days, you've made the mRNA and you're off to animal studies. So those would be three fundamental reasons why it's such a powerful technology.
Wow. So let me just build on that. Dr. Kahvejian, I just want to bring you back in. If we sort of go beyond COVID and think about mRNAs in sort of a broader spectrum, what does the success of the COVID vaccines imply for how we might make use of RNA in the development and discovery of new drugs? Or maybe I'll put it more bluntly to you, do these discoveries revolutionize the way we think about how we treat disease and illness in the future?
Well, at Flagship and at Moderna, we've always been firm believers in the vision that a programmable molecule like mRNA and RNA and even DNA could be widely applicable to a wide range of diseases. And what this demonstrated was that indeed you could put that into practice. You could encode something very quickly. You could make it. You could deploy it. That it would be safe in hundreds of millions of people. So it is, yes, a big watershed moment in demonstrating that, but the programmability of the molecule implies that you could use it for a wide array of other infectious diseases, of course, but also in other areas. So if you imagine that these molecules can allow us to make a therapeutic inside the body, then by choosing the sequence, you can have the body make whatever protein you want. And a wide array of diseases today are treated using proteins. We make the protein outside the body and we inject it into the body. So if you can have your body make it itself, it opens up a lot of opportunities and accelerates things dramatically. But of course, all of that will require more research. It'll require more time, but we're really excited about it. And Moderna and other companies are thinking along those lines. Even though the COVID product and the COVID vaccine has sort of overshadowed everything because of its impact, there's a lot going on in this field that maybe few know about.
Mm-hmm. And we definitely want to get to that during the time we have today, and particularly the issues around cancer and immunotherapy. I do want to bring in Professor Kahvejian for a different perspective on this. But I have one more question for you, Dr. Afeyan. I'll just put it very bluntly. Are you happy with how the Moderna vaccine has performed in terms of preventing illness and hospitalizations and deaths from COVID?
I mean, these are, of course, we're happy to see the science translate into medicines that have global impact. This is why we all do this, and we all do it whether it has impact on one person or a million people. And the difference between the two is actually circumstances, I would say, as opposed to something we do a lot more of. I mean, in this case, there was quite a lot of heroism involved at the level of Moderna's team to be able to scale up production of this class of molecules that has no precedent to the kind of doses we had to get to be able to supply enough. You have to imagine in 2020, when we started, I think we had historically made but a few thousand doses to try in human trials. To go from that to 1 billion doses that we made plus or minus last year since then is quite the scale up. So, it's less glamorous because the science, of course, is where a lot of the magic resides. But actually, in terms of the remarkable feat, I'd say that probably is what's made me more gratified. That it was safe, that we could bring a lot of different government, non-government constituents into an alliance with us globally to test and to deploy these things, get it into arms. I mean, that has been like this. There's probably 10, 20 levels of heroism involved, most of them away from us, actually. And so we are very gratified that things worked out. There's probably a whole lot of ways in which things might not have worked out, even if the science worked. And I'm mostly happy about those.
Mm-hmm. Wonderful. I can imagine how much paperwork was involved in that as well. But so I suppose one other sort of important piece of this equation on top of these incredibly so the emerging technologies is the cost involved in all this. So I do want to bring you in, Professor Kinavale. You're the Canada Research Chair in Policies and Health Inequality, and I want to get a sense from you of how these advances might change how we think about access to top-of-the-line healthcare, especially here in Canada, where we pride ourselves on health equality, or so we say. So let me break this down into a two-part question for you. So firstly, is the scientist in you impressed with how quickly the biopharmaceutical industry has managed to turn our understanding of our mRNA molecules into a safe and effective vaccine against COVID? But secondly, is the sociologist in you concerned at all about how do we get populations, from less wealthy communities and countries access to these same benefits?
Thanks, Derek. Well, to answer your first question, I think it's important, as our colleagues just pointed out, to remember that these mRNA vaccines may be new, but using mRNA for these purposes is not a new idea. And in fact, it owes a lot to fundamental academic research, such as Jerry's work, as was done at McGill. And this was way before it was picked up by pharmaceutical companies as something that could be marketable. So we really have our universities and our funding agencies, like the Canadian Institute for Health Research. And here, full disclosure, I'm on the board, so there is that little bit of conflict of interest to disclose. But we really have those institutions to thank for the initial research that led to these amazing feats of medical science first. And, as Noubar pointed out, I think for me, the unsung heroes here are also all the scientists that work with, sometimes volunteering. I mean, the amount of work that went in to standing committees reviewing the evidence, the safety, efficacy data that was pouring in a matter of weeks, hours, and turned them into recommendations for our regulatory agencies like the FDA and Health Canada. That's the amazing feat to me, that all of that aligned. And then finally, that our governments took a leap of faith and that they would buy these vaccines sight unseen, even before they, or put money towards them, even before they were produced, and the populations lined up. I think that's what really amazes me. And it's the fact that all these trends came together that's impressive. Sociologists, social scientists were systems thinker, and this was the alignment of systems, of course, for an existential threat in a way that we'd never seen before. And I think that was truly, truly amazing. Now, turning to your second question about equity, I think that a big roadblock, as you hinted at, that we managed to avoid in Canada is what we call the inverse care line healthcare services research. And this expects that people who need care the least will get it first. Usually that tends to track with money, power, connections. And so these are not necessarily people who need it medically the most, but typically it isn't. Now, I think, thankfully, our government and our population in Canada are pretty staunchly committed to equity principles, generally speaking. And the threat was so existential that the government just put such strong gatekeeper policies in place that ensured that really people most at risk of complications would get the vaccines first. So I think we should really be proud as a country of how disciplined we all were in waiting our turn. And that this ordering was based on epidemiologic and public health recommendations. So that, to me, is also fantastic that it really came together. But that doesn't mean that we didn't have fault lines, the cleavages that had been growing over time. And so we have groups in Canada that have good historical reasons not to trust the government, and especially medical interventions. And this includes some Indigenous peoples, but also racialized groups, and even more true in the States. So when we're thinking about developing medical interventions, I think we need a way forward where we bring all groups that are affected by these interventions together, hear the diverse voices so that their perspectives are also taken into consideration. We can't just leave people behind and say, well, they don't understand or they're listening to the wrong messages. We need to do better than that. And we learned that from COVID. And all of that encapsulates global health, because those feats of medical science, these particular vaccines that we're talking about, they weren't designed to be delivered in resource poor settings. The cold chain. I recall that Ontario even struggled, in the dead of Canadian winter to deliver some of these vaccines to long-term care facilities that didn't have the capacity to maintain the cold chain. So imagine in sub-Saharan Africa. So I think those are the challenges that we're facing, because as we've said time and time again, none of us are truly safe from this virus until we're all safe.
Right. No, that's very interesting. So I'm wondering, Dr. Afeyan, from Moderna's perspective, what is sort of your perspective on sort of access from a sort of global health perspective? So, for example, if we think back to the COVID vaccines as an example, we saw, countries like the United States and Canada, hoarding supplies of product or securing an oversupply of contracts. And yet citizens in Asia, Africa, Latin America often had to wait months and months just for their first shot.
Well, I think that the fact that pandemics happen once every 100 years means that we were completely, as a globe, unprepared for the logistics involved and the decision-making that had to go into this. Second, I'd say public health is perennially an under-resourced and underappreciated and undervalued part of our health a net, if you will, that humanity really relies on. And at times like this, they don't have the standing to basically, step into the breach and actually lead decision making. And so it ends up becoming the purview of many other factors, including politics, which we saw rampantly. And of course, the worst kind of that is kind of national or regional politics that ends up favoring one population or another. I think that's been well covered. As a manufacturer of an innovative solution and as the pioneer of the space in general, having spent 10 years building the platform that's ultimately been used by anybody who's doing mRNA vaccines in this space. On the one hand, we were gratified that we had others get into the space so we could have enough supplies. But then when we realized that our own supplies would be essentially commandeered to be directed only to where people could kind of had pre-ordered and prepaid for, that was not a comfortable situation for us, because there's exactly nothing we could legally do about it, and we said so. On the other hand, of course, people don't want to hear that. They want to blame people, and so Moderna and Pfizer received a huge amount of blame for not being able to get vaccine to the rest of the world. Of course, the solutions that were proposed, which have been well covered, was that somehow overnight people would start manufacturing these things and put them into people's bodies at all corners of the world. It's a completely impractical, kind of from a safety standpoint, impractical solution. What was a solution is that we and our other colleagues in the pharma industry deployed billions of dollars to ramp up production so that we could actually adequately supply and in fact oversupply the vaccine, such that last fall, when there were concerns about the African continent not having enough vaccine, we were getting reports from the continent saying we're getting barraged with vaccine, and within 3 months the news cycle shifted to they have way too much vaccine, and what they didn't have, which was reality, is the ability to get into people's arms, the education, the supply, the cold chain, the logistics. And frankly, that's a place where I think pan-government collaboration could have well smoothed the way, but everybody was so panicked and so beholden to their own populace, which was stuck at home trying to go out and walk their dog and being forbidden from doing that. As you guys well know, in Canada, some of the curfews that were put in place, it's just human nature. These things took over, and there's no excuse for it. But at the same time, I think we all have to look at our own operations and say, what can we do better? In the case of Moderna, we've been pretty public about it. In October of 2020, we publicly disclosed that we would pledge not to enforce our patents globally during the pandemic for anybody using our technology in mRNA to make other vaccines. And we have stuck to that pledge. We've expanded that pledge now to include permanently low-income, low to middle-income countries, not just during the pandemic. We will not enforce our patents there either. We further announced, and has been recently much more specific, that we will build a plant to make mRNA vaccines in Kenya for the continent. But importantly, we will fund it. We're not looking for any money. And we will make sure the expertise is there to make safe product, not just for COVID, but for any number of vaccines. And that creates what I'd call vaccine security as opposed to access only. Because if you have the ability to make it, you want to make it to world standards. You don't want to say, well, I can regionally make something and let me try that. And so we're fixing that. So we'll do everything we can. But at the end of the day, as Amelie mentioned, this is a complicated social and logistical challenge. And you know, it really requires collaboration. We saw unprecedented collaboration in the last 2 years, but it turns out no amount of collaboration proves enough to solve the whole world's problems. And this is going to be a test run, frankly, for climate change. I will tell you that I'm sure something else that McGill's heavily involved with. If we don't get this right, I'm a little discouraged about what we're going to do with climate change, because it's going to feel the same. There's going to be a rampant panic as these things start hitting us in a more severe way, and everybody's going to start protecting their own, and it's going to be really challenging. So I hope we come out of this pandemic and other ones like this with some lessons that can apply beyond just this space.
Very interesting. Wow, very thoughtful, very interesting. So let me go back to you, Dr. Pelletier. I'm just curious, you talked, Dr. Afeyan, about collaboration. So I'm curious, from your perspective, Professor Pelletier, someone, again, who spent your entire career working in this field and mRNA technologies, do you feel that in some ways the pandemic has perhaps helped accelerate some of the research, whether it, you know, speaking of more investments coming in from government or private support, and perhaps even accelerated some of the collaboration that may not have existed amongst rival teams and rival laboratories?
The short answer is for sure, yes. The Canadian government did put up a special call for proposals around SARS-CoV-2 when the pandemic emerged. Everybody wanted to see a solution to this problem ASAP. And then you also saw the emergence of scientific forums and partnerships that fostered exchanges of ideas in Canada. You also saw this in the States with Operation Warp Speed. You saw Moderna collaborating with NIH, and you also saw CDC and the FDA coming in and ensuring input into clinical trials and ensuring that the data was reviewed. The data that emerged was reviewed as quickly as possible. So these were associations or collaborations that had not previously been explored. So yes, you definitely saw new mobilization of effort across agencies. But remember, scientists by nature, I feel, are quite a collaborative group. And so when a new problem like this emerges, it's quite natural for people from different expertise to get together and try to solve it in a collaborative manner.
Okay, wonderful. Thank you for that answer. So just a reminder, we're starting to get some questions that are coming in from alumni. If you have any questions for us, you can send them to us by email at aoc@mcgill.ca. Like I said, we're getting a few, so I'll try to go through and pull out a few that make sense. Let me just, let's look to the future for a minute before we get to some of these alumni questions. And maybe I'll turn to you, Dr. Kahvejian. I know that you've done a lot of work around, you know, obviously the RNA platforms, mRNA, but also getting into things like artificial intelligence and machine learning. So I'm wondering, what are some of the lessons that we may have learned from COVID and developing the vaccine that might be applied to future drug discovery? So, you know, is this, you know, AI, is this the next frontier in biopharmaceuticals? And what might it mean for patients and patient care in the future?
Well, that's a pretty broad question. I'll try to maybe relate it to RNA in particular. As we discussed, RNA is a programmable molecule. It's a string of letters, A, C, G, and U, combined in different ways to make different kinds of proteins inside the body. The way we put those letters together has an impact on how much translation we get, as Dr. Pelletier mentioned, how stable the RNA is, how immunogenic it may be. And so that's one example of an area where machine learning could be very, very powerful, and where we're applying it to understand how that sequence variation relates to all of those different properties. That wasn't possible before. When I worked at Nahum's lab and when Jerry worked in Nahum's lab, we would try a set of sequences and see what happens, but that would only be one example. It would be one sentence we would put together. Now we can imagine putting millions and millions of combinations together and then using machine learning to elucidate the rules that governs sequence to structure, structure to function types of relationships. So there's a big acceleration in our ability to now design medicines. We're now going from an area or era of just trying different medicines to designing medicines in a much more deliberate and engineering kind of, with an engineering kind of mindset. So that's one way AI and ML are impacting all ranges of therapeutic discovery.
Now, Dr. Afeyan, I know I was reading some of your writings recently. You talk a lot about digital biology and how this brings medicine and drug discovery in particular from sort of taking a lot of shots on net, sort of in the hockey euphemism, to being much more precise and much more scientific. Can you talk a little bit about this notion of digital biology and what that's going to mean for drug discovery in the future?
Sure. I mean, you know, it hasn't been that long that biology has become a molecular science, and even less time since it's becoming an information science. And we've known that DNA and RNA are codes for life, but other than that metaphor, we haven't been able to treat it like much of a code, because we haven't understood what the code stands for. By and large, I mean, we know that DNA codes for RNA, we know that RNA codes for proteins, and then we know that proteins do stuff. But the ability to then program at the level of DNA or RNA and be able to affect end results in disease or cellular processes, that's not a very long-standing capability. And so we view the real fusion of a digital set of technologies, not just to measure or to handle the data, but really just the way to think about what we're doing and biology, as a very exciting kind of new field. We have to be very careful because it's not a direct analog to software and computing and stuff, but more and more there are ways to think about it in that way. So we view the benefit of all this as what we call programmable medicines. I mean, if we could think about programming of a code that can instruct your cells to make any protein you want, any combinations of protein you want, or that protein further programming your immune system to do what you want, that mindset, the reusability, the modularity, the recombinability of the elements to get different instructions, I mean, these are exciting things. It used to be that the cycle time in molecular cellular biology experiments was such that, you know, it would be like, hurry up and wait. I mean, you could design something and then 6 months later, you would get the reagent needed to do something. Now you could do it in the next minute, in the next hour. And so what questions can we ask by being able to make these code molecules? It's interesting. It goes back to a very famous book that Schrödinger, the famous quantum physicist, wrote, called What is Life? And it was his musings as a physicist, theoretical physicist, you know, some in 1943, about how a physicist viewed life. And he there describes this notion that life must be based on a code script. Well, code script as a notion hasn't really been an actionable mindset within medicine until very recently. And I think mRNA gives us a mechanism for that. Certainly viral delivery of genes begins to do that. And I think we're going to be in an era where those two fields and all the techniques we have with machine learning, which can really get a lot of simulations, if you will, done, and learning cycles done, all of that is on the future. And so it's great for people who are where I was in my career 40 years ago, or Dr. Peltier may have been, and others. I wish we were in the beginning, because it's going to be an incredibly different cycle going forward.
Yeah. So let me ask you, Professor Kahvejian, how do we train people, the next generation? You must see lots of McGill students coming through. How do we train them to be the leaders in this kind of new approach to medicine that Doctors Afeyan and Kahvejian are talking about?
I think the key is bringing people together. As I said, for different communities with researchers, not just post delivery, but also as it happens. So we need to get people to be nimble, fast thinkers, bridgers across processes, across disciplines, and across sectors too. So in academia, at least, especially in arts, let's say, but also in some disciplines in medicine, we train for what we know. That is to say, most of us have been in school all our lives and we just keep going. But we also need to just make the students aware that there is such a wealth of opportunity out there for them that goes way beyond academia, and data-driven decision-making, which is some of what we're talking about here, it happens everywhere. And the more that happens outside and understandably to people who can connect also with academic ideas, the better the better off we will all be, all our society. So I think that's really it's something that we do in social science, but it's also happening now increasingly in medical sciences. We need to bring people together.
Okay, sounds simple and complicated at the same time.
It takes a lot of work. It's a budget line and it's a process.
Yeah, and Derek,
Right.
what we're doing in industry is obviously we're realizing we have to cross-train people. We can't the departmental model in universities is good for certain things in helping drive specialization and disciplines in certain disciplines, but when it comes to doing things like what we have to do to bring a new kind of medicine to humanity, it takes a lot of interdisciplinary work. But even bringing people together requires them to have some common language and an ability to converse with each other and understand each other's sciences and each other's qualities and how they can contribute to something together. So there has to be quite a bit of cross training and probably maybe new departments or new ways of teaching at universities to allow for that cross training.
I think, if I may jump in, increasingly, universities are no longer in the knowledge giving kind of business. They need to get students to understand that knowledge is out there. They need to be able to assess it critically, assess it. Yeah, that's what I said. Assess it critically and be able to then write and speak argumentatively. And I think those are the key skills that we need to teach. And that is what brings people to be able to then jump from discipline to discipline, to understand each other and to bring those connections together.
Okay, wonderful. So I'm about to jump to some of the questions that are coming in from alumni. But before I do that, last question for you, Professor Pelletier. I'm wondering if we could talk a little bit about the broader implications of mRNA technology, specifically on cancer. I know that's one of your areas of specialty. And I've read lots of articles in the last number of years about immunotherapy, perhaps replacing radiation therapy or chemotherapy as the next therapy of choice for certain kinds of cancers. So I'm wondering if you could talk a little bit about how do mRNA molecules, what role do they play in essentially turning our bodies into our best weapons to fight cancer? How was this working, and have we seen any promise to date?
So you're absolutely right. There's an enormous amount of excitement in immunotherapy. There's currently 3 approaches that I can see where mRNA therapeutics is being used to try to harness the body's immune system to combat cancer. One is to deliver an mRNA molecule that will teach the body to make antibodies against a protein that is mutated, frequently mutated in certain cancers. And so this would essentially result in the body's immune system seeing that tumor cell as foreign, seeking it out, killing it, and eliminating it. So that's one approach that's being investigated. The other one is it's emerged that tumors will often create a dysfunctional microenvironment, which helps them evade the body's immune system. So they sort of build a shield to protect them against our immune system. And so one can deliver protein molecules via mRNA to disable this shield and now make the tumor visible to the immune system. And that's a current path that's being tested in clinical trials through the mRNA therapeutic platform. And the third way I can see is many people, I'm sure many of your listeners have heard of CAR T cell technology. It's a technology where one isolates immune cells from the body, manipulates them in the lab, and then re-injects them back into the body with the added feature now that these cells can recognize tumor cells as being foreign. So they seek out the tumor cell and eliminate them. The problem is, one, this manipulation in the lab is quite expensive, and two, these CAR T cells actually stay in the body for several years. And so they can lead to some downstream undesired effects. With mRNA technology, what's quite exciting and has recently been shown is you can actually inject the mRNA directly into the body. The appropriate T cells will take up that mRNA, become modified to kill the tumor cell. And because the mRNA has a short half-life, as Avak mentioned, it's gone within a few days. So then that modified T cell goes back to normal, and you don't have this long resident time of these modified cells in the body creating downstream problems. So these are three ways in which the technology, as far as I know, is being applied. And these are actually, there are clinical trials now ongoing with some of these approaches.
And is there any sort of specific forms of cancer where you actually see this having, effective treatments being developed?
The type of treatment being applied will depend on the cancer type. So some cancer types express some of these peptides that mark them as being foreign more frequently than other cancer types. I'm not so familiar with the clinical trials as to tell you, this particular cancer versus that one. But the immunotherapy one, actually, the immunotherapy and the CAR T therapy has the potential of being applied across a broad range of tumor types.
Okay, interesting. So we have about 15 minutes left. Let me turn to some of the questions that have come in from alumni. Maybe I'll put the first one to you, Dr. Afeyan. It comes to us from Vivian Krieger. She's asking, what is the turnaround time between identifying newly highly infectious variants and the production of new mRNA vaccines? And how onerous might the approval process be with newer, slightly modified vaccines?
Well, the cycle time of having a new sequence that we could generate in quantities enough to test it in humans preliminarily is extremely fast. It's days. And then the question becomes, what do you have to show in order to have it deployed? At the moment, it's kind of somewhere in between a brand new vaccine and the same vaccine. It's kind of in the middle, where you have to show obviously some level of safety and just making sure that you can make it in animals and show some safety. But you can pretty quickly get into humans now because there's such a database of experience with mRNA. And unlike proteins and attenuated viruses, each one of these looks very, very similar to the previous one, except for the antigen that it codes for. And so I think that right now, as we just disclosed, we showed data in a trial for our bivalent vaccine that is designed for the fall and has shown quite broad coverage of Omicron and all the other strains. And when from the time we had that bivalent vaccine ready to try till now, it's probably 4 or 5 months. But at this point, we have to go through the regulatory review, and we're quite hopeful that we will be able to be in a position to supply it before the fall, because we think that coverage of the broader range of variants is going to be quite important.
Makes sense. All right. Our next question is from Marty Fisher. You might want to jump on this one as well, Dr. Afeyan. So he's asking, how soon are we expected to have an effective mRNA vaccine for the seasonal flu? And in the event that this does happen, is it possible to combine a flu mRNA vaccine with a COVID vaccine and provide one combined vaccination each fall?
It's a good idea. It's such a good idea that we've already been working on it for a year, and we are in human testing, advanced human testing, and our hope is that by the next season, that is not this fall, but the next fall, there will be exactly that on the market. Flu is a bit of a different situation because there are many, many strains that exist simultaneously, and it's always this mystery as to which strains we might get for the next season. And in fact, we're just now in the process of kind of determining what the seasonal flu kind of would have to look like, and we'll be able to test it in the southern hemisphere separately and in the northern hemisphere. So there's some interesting learnings that we can do by testing it around the globe. But basically, yes, we think that we'll be able to, again, we're testing it, have a flu version and a combination. Plus, on top of that, we're working subsequently to add an RSV vaccine as well into the same. These are all respiratory vaccines, and we think from a compliance standpoint, getting 2 shots once a year of these will be the most appropriate level of protection, but we have some work to do to add that third one as well.
Okay, great. Okay, let me direct, maybe Professor Pelletier, I'll direct this next question to you. It's scientific with a little bit of a political spin. So, the question comes from Claude Lalonde. He's asking, are there any plans to use this mRNA technology for a preventive or curative vaccine for HIV? If so, what is the expected timeline? And is there the same political will for the HIV/AIDS pandemic as there was to make it happen fast for the COVID pandemic?
Good question. So let's not forget, we've been in an HIV pandemic since 1981. There's been a lot of effort dedicated to developing vaccines against HIV, which have fallen short of the mark. Having said that, I believe NIH just launched a clinical trial this year of 3 mRNA-based HIV vaccines. We know that control of HIV is possible in infected persons because there are these HIV-infected individuals who are known as elite controllers. They basically are infected with HIV, but they're able to live with the virus for many years without any immune damage. And if we could understand what the molecular basis of this elite control mechanism was, definitely I do think the mRNA platform technology could be harnessed to take advantage of this mechanism. So HIV is also a more complicated problem than coronavirus. You know, I'd say HIV is a PhD level virus, whereas coronavirus is, you know, not to downplay or minimize the impact that corona has had on health worldwide, is a much simpler virus compared to HIV. In fact, people infected with HIV have antibodies against HIV. That's one of the ways you know they're infected. So the mechanisms will be a little bit more complex. There's nothing, however, that I can see that's beyond the realm of mRNA technology that can't be used here to one, try to immunize, and two, those individuals that are infected, try to control the infections a little bit better.
Okay, great. So we have about 5 minutes left. I'll try to race through as many of these questions that have come in. We got a bunch of questions from Amal Luji, but I'll focus on one. I'll put it to you again, Dr. Afeyan. So he's saying that, well, I was going to ask you about this, that, you know, Moderna's recently announced plans to build a vaccine plant here in Quebec. His question is when, where, and why Quebec?
We undertook a thorough review of the various alternatives against both short-term, long-term considerations, because these are what we call resilience plans. We're trying to build these with a view towards kind of a decade or two forward looking, what vaccines we may make there and how they could be used for dual purpose in case new pandemics show up. And so at the end of the day, based on that review, Montreal was chosen to be the appropriate site. We're also expanding our research presence in Canada, and we'll also be doing quite a lot of collaborative work. We also have the academic access program that McGill was the first to avail itself to. So I think this is kind of not a one-shot thing for us, but it, you know, it was just done in a highly professional way, and the conditions were viewed to be most suitable to do that in Montreal.
Mm-hmm. Now, you're obviously based in Boston, where you've got the incredible network of universities and a burgeoning biotech field. I guess in many ways, Montreal would be very similar with the universities we have here, including McGill.
Yeah, I think Montreal has a lot of advantages, and I think the Canadian government has been, you know, broadly collaborative and supportive of building kind of an ecosystem there. Obviously, there's other areas as well in Canada, but I think the academic strengths, certainly in McGill and elsewhere, coupled with translational work in the hospital research environment, coupled with companies, is what you really need to build a cluster, and you know, hopefully Moderna's presence there will benefit that.
Okay, great. Okay, Dr. Kenavale, maybe I'll throw this next one at you. It comes to us from Genevieve Snyder. She's saying that during the pandemic, there was amazing and selfless collaboration to quickly find solutions to combat global pandemics. When there is not the urgency of a pandemic, what are the main roadblocks that prevent such global and rapid collaboration?
That question brings to mind something that Ed Young, the Pulitzer Prize winning Atlantic author, said. epidemics and tech solutions flow in opposite directions. So epidemics flow downward into societies, into the cracks, target the most deprived. While tech solutions, they flow upward even when they're rolled out in universal systems. They're picked up first by those in most privileged positions, with more education, power, connections. And once that's fixed, they move on, right? When they get that service, they move on. And so I think what we need to bear in mind is that inequalities tend to persist even when the disease that produced them, that's one of the reasons why the inequalities persist. So I think that we have to make sure that we bring social science in with the medical science, because if medical science gave us those vaccines, we have needed social science and public health science to get them into people's arms, understand what are the roadblocks. So we need to move beyond the cycle of panic and neglect that has plagued public health, as Dr. Afeyan said, and really think about what are robust systems, bringing those communities together. I keep saying that, but bringing the communities that are seldom heard to the forefront, and hearing their perspective in a continuous fashion, not in a like, oh my God, this is an existential crisis, let's move ahead. So I think we need to build the platforms, and we're fortunate at McGill that we have the capacity and the interest to do so.
Wow, this is certainly sounding like a big advertisement for McGill. That's a good thing. I guess it should be. Let me try to squeeze one last question in. This one comes from Douglas Yip. I actually think I know the answer to this, but I'll put it to you anyway, either Dr. Afeyan or Dr. Kahvejian. How is it that Pfizer commercially developed an mRNA vaccine that seems to have predated Moderna's? Did they have a parallel mRNA development program?
I'm happy to answer it ever so politically correctly. Pfizer partnered with a German company, BioNTech, which had an mRNA program. That mRNA program was entirely focused on cancer up until 2019. In 2019, I think they announced that they would work together with Pfizer on flu, and the pandemic hit several months later. Now, I think it's fair to say that Moderna's involvement, having 9 human trials in vaccines in the clinic, may well have been useful for any new entrants in the area, in the sense that there was a body of work and a whole vast set of disclosures, both in patents and papers. And I've said publicly, I think it's good that was helpful for others to get involved. The program that Pfizer had, they've never claimed predated Moderna by any shot. What it did do, and which may be interesting to your audience, is that they were able to get to the finish line 2 weeks before Moderna did. What is forgotten, however, is why that happened. Moderna entered the clinic a month and a half earlier, got first phase 1 data announced earlier, and then in the middle of 2020, when we were doing a 30,000 subject study, we realized that, in speaking about equity, we realized that the trials were enrolling, both for us and for them, largely white middle-aged men and certainly of a certain income level, just because they were much more used to going to trials. And we realized that we had one of two choices. Either we would make a study that completely consisted of inadequate diversity, or we would slow down, shut down some of our sites, and insist on having more representative diversity. We did the latter, and we lost a month in doing so, and we ended up finishing 2 weeks later than they did. It's in the historic way, it was the right decision. It was one of the most painful decisions we made. But it is remarkable. I was just in DC in the last 2 days, and I now realize that there's legislation afoot to force companies to do that. It's the right thing to do. They shouldn't have to be forced. But in our case, we did it in the most visible and only product that we ever developed. And I'm very proud of my colleagues to have made that decision. But that's why they ended up seemingly having started earlier. In fact, they started quite a bit later, but we slowed down.
Wow, wonderful. That's exactly how I would have answered that question. What a great way to end this as we wrap things up. So I realize we are just about out of time. So we are going to have to wrap up the conversation for today. Before we close, I'd like to remind you that this video will be available at this very same link after our recording ends. So feel free to share it with others who may not have been able to tune in live. And of course, I'd like to extend my deepest gratitude to our 4 wonderful panelists, McGill graduates and biotech executives, Noubar, Afeyan, and Avak Kahvejian. and Professors Jerry Pelletier and Amity Kenavade for joining me today to discuss such a fascinating topic, and one that promises to have broad implications for how we confront and battle disease and illness for decades to come, and really giving us a little peek under the hood, I think, in some cases, to what went on behind the scenes at Moderna in those first few frantic months when the pandemic began. To all of our viewers, please continue to stay safe and be well as we navigate through what we hope is the final stage of this COVID pandemic. And please continue to stay tuned to your McGill email and social media feeds for more updates on alumni webcasts and more exciting programming from McGill, both online and in person. Thank you. Thank you, everybody.
Thank you.
Thank you.
Thank you.